Does TMAO actually cause heart disease, or is it just an innocent bystander? TMAO is a metabolite, a molecule present in some foods, and it can also be produced in our body. And TMAO stands for trimethylamine N-oxide. It's basically a nerd's wet dream. And one of its precursors, one of the things that can be converted into TMAO, is choline, which is a nutrient found in many foods. Eggs and liver, for example, are rich sources of choline. So in our gut, in our intestines, our gut microbiome can convert choline into something called TMA, which then gets absorbed, and in our liver finally gets converted into TMAO. Another example of a precursor is carnitine, which is high in foods. For example, red meat has a lot of carnitine, and so that can also be converted into TMAO in our body through the same sequence of steps. And finally, some foods already have TMAO preformed in them, and fish is one example that's high in TMAO. There's a lot of research that people who have higher levels of TMAO in their blood tend to have higher risk of disease, like heart disease or diabetes. But that doesn't mean that it's TMAO causing these diseases, right? It could just be an innocent bystander, a marker, and something else is causing the disease. There's also lots of data in test tubes and lab experiments and mouse models revealing lots of physiological effects of TMAO in cholesterol metabolism and traffic, inflammation, oxidation, etc. So I've basically held every opinion on this. I used to be more worried about TMAO back in the 2010s as the evidence was mounting, pointing to some concerns. Then a series of studies came out starting in 2019 suggesting that TMAO might not actually be a cause. And so that changed my mind a bit. And I've generally leaned more towards the innocent bystander scenario. So I recently spoke with Dr. Kevin Klatt, who is a registered dietitian and also has a PhD. And among many other things, he has researched choline metabolism. And Kevin is one of the most rigorous and one of the most objective scientists on social media. Highly recommend following him. I always learn a lot by following and reading his content. So we talked about all this. Kevin made really good points, evidence-based points, and I changed my mind a bit. Again, I'm less sure about the innocent bystander scenario, and I'm a bit more undecided again. Our conversation did get a bit technical, but I'll jump in every few minutes to summarize or to explain any terminology, so please bear with us. And we did wrap up with a take-home message that I think makes a lot of sense. If it helps, you can turn on the closed captions because we did speak a bit fast and there is a lot of jargon. But I think it's good for you guys to see a conversation that isn't all black and white, where we're considering a number of facts of evidence and where there is a significant level of uncertainty because that is a big part of science. So here we go. Enjoy. What's your general take on this whole story of TMAO? Do you think it's a cause of risk or do you think it's an innocent bystander?
I think it's complicated. I would just say at the top, we don't have cardiovascular outcome trials reducing TMAO. I know that there are some specific inhibitors to prevent the microbial generation of TMA to try and lower TMAO that are under study, and I don't think we have the results of those yet. At least as of today, February 2024. Folks will listen to this in 2 years. Don't crucify me.
So Kevin is referring to randomized trials looking at things like heart attacks. That's what cardiovascular outcome trials means. So giving people something that inhibits that microbiome conversion that ultimately leads to TMAO production and seeing if those people have less heart attacks, for example. So that type of experiment in a randomized way is the highest standard of evidence. That hasn't been done, so we're less certain.
But yeah, so we don't have cardiovascular outcome trials. As you mentioned, the Mendelian randomization comes up. I'm pretty dubious on Mendelian randomization. I think it's really helpful in some instances, and I think in other instances it gets quite abused. When you look into there's a couple of Mendelian randomization studies. One finds it largely relies on an FMO3 variant, that is the enzyme that converts TMA to TMAO. They do find increased risk of cardiovascular disease with that. But FMO3 has other substrates besides TMA, which is the sort of the asterisk there to say maybe this isn't the perfect inference around TMAO that we want it to be. And then FMO3 is also regulated by insulin signaling and a whole bunch of other stuff, and it gets really complicated.
So Kevin is talking about genetic studies. That's what the Mendelian randomization is referring to. So these are studies where you look at people who naturally carry different genetic variants, different forms of the same gene. So for example, different genetic variants of the enzyme in the liver that makes TMAO. The problem is that same enzyme also does a number of other things, so it gets a little complicated. So for that reason, these specific studies are inconclusive.
On the other side, there have been other variants associated with TMAO that don't associate with cardiovascular disease risk. I think a lot of them use, if I recall correctly, are assuming a totally linear relationship. And that's also something I'm not, from the epidemiology, it looks linear, but I do think that this could get quite complex and there could be more threshold effects that are apparent that are not going to pop out in. So like you could look at the lower end of TMAO, like below 10 micromolars, and see that 8 does worse than 2, and that could be because people with diabetes or people with kidney disease or early kidney disease are showing higher levels, and it could totally be confounded. But at some point along that spectrum, when you get up to like 50 micromolars, there's at least some in vitro evidence that you're activated the unfolded protein response. So it's a TMAO binds PERK at 50 micromolars in culture and activates that cell stress signal. And so, at some point along the threshold, I think there is potential evidence for TMAO to causally cause cellular damage. And so I don't want to rule it out, but I think this is going to be complicated of you have to find people in a true causal risk threshold where TMAO is actually bad and then lower it. And so I will wait for the cardiovascular endpoint trials really to be the more definitive thing on this.
So Kevin is talking about different ranges of TMAO levels in the blood. There are some genetic studies that suggest TMAO might not be a cause of disease because people with a bit higher or a bit lower levels of TMAO don't show significantly different risks of disease. But what Kevin is pointing out is that maybe at a much higher level of TMAO, we might still see harm. So that's a possibility.
When you go to just the observational epidemiology, I think it's super clear and relatively linear that higher TMAO levels are associated with higher incidence of adverse cardiovascular events. And that is observational. I think there's a lot of the data is different cohorts adjust for many different things. So TMAO is a product of not only making TMA from dietary precursors, but the liver has to turn it into TMAO. And so that is regulated by insulin. And we know diabetics have higher TMAO levels. We know the hepatic insulin receptor knockout mouse has sky-high TMAO levels. So insulin's definitely regulating that. So there's a non-dietary factor that definitely comes into how much TMAO is made. And then you have to be able to efficiently pee out the TMAO. And so folks with chronic kidney disease or other kidney dysfunction can exhibit, it looks like they have a buildup of higher levels of the plasma because the kidney is less efficiently clearing it. Like when you look at some of the choline feeding study stuff that's been done, you can definitely see a postprandial rise in TMAO, but it all ends up in the urine, and the fasting TMAO levels typically aren't much higher. It depends a little bit on the study and the form of choline that they're using, but a lot of it gets peed out within healthy populations, which is what we've known of TMA and TMAO metabolism for years going back. And so the epidemiology, I think most folks agree it's at high risk of confounding by metabolic status and kidney function. And different cohorts will find different things depending on how heavily they adjust for it all. So
Kevin is pointing out caveats of observational studies where we just measure people's TMAO level in the blood, and then we look for a correlation with the presence of disease. But the problem there is one of the problems that people might have a higher level of TMAO because they already have developing disease underneath, like diabetes or prediabetes or kidney disease, and not the other way around, right? So this is what we call reverse causation. So the genetic studies can help us get around this reverse causation to some extent. But what Kevin is also pointing out is that because TMAO can come from food, but it can also be produced in our bodies, somebody with a higher level could be eating more of certain foods, could be producing more in the body, or they could be excreting less. So this always has to be taken into consideration.
It's not a perfect world. I would put my bets on a cardiovascular outcome trial with something that inhibits TMAO production or TMA production in the gut microbiome is probably going to see reduced cardiovascular events, particularly if it is in a really high TMAO population. I would much less hedge my bets on if you did a choline restriction trial in a population like, is that going to reduce cardiovascular events? I don't think we will ever do that trial because you would probably need hundreds of thousands of individuals to do it. The other thing I didn't say too is there looks like there's a microbiota composition that's more prone to making TMA versus a microbiota composition that's less prone. And so there's still a lot of work going into that space. So if a disease is making your microbiota more TMAO production prone, that again is another factor that's going to influence what your circulating TMAO levels are. So anytime you look at a metabolite in plasma, you have to think of how it's homeostatically regulated. And there are, just because nutrition can affect it does not mean nutrition is the primary determinant of the circulating levels. And so when you look at some of the data, only one real study I know of this out there that looks at like the percentage of variance in TMAO levels, it seems to be explained by variance in diet. And it's only like 17%. And it's largely, it's like animal foods, but it's not clear that like dietary choline itself is really the major explanatory factor, largely because there's other things that affect it. So there's like carnitine is also a precursor. Red meat itself seems to not only provide precursors, but also influence kidney handling and excretion of TMAO. Stephen Hazen, who birthed the whole TMAO story, has also done a lot of that work. And then fish is like the big outlier. So, we've been recommending fish for forever. And I know there's a lot of people who are super enthusiastic about like, essentially the conversation we have is TMAO a surrogate outcome that should guide dietary guidance? And much like we use LDL cholesterol to guide dietary guidance to reduce cardiovascular risk, should we be using TMAO that way? And a lot of people are really enthusiastic, and a lot of like the plant-based community is, they're like, yeah, this is like another nail in the coffin for animal foods. But there are now several controlled trials showing that like whole grain interventions increase TMAO, fish interventions increase TMAO, and things like there's been some fermentable fiber studies that have shown increased TMAO as well. And we look at population studies, like the predominant predictor of TMAO is your fish intake from the diet. And so we have to walk back decades of dietary advice and give a big whoopsie if we really are going to start using TMAO as a surrogate indicator. And again, fish, despite being a massive source of preformed TMAO, and there's a lot of variation in the type of fish, but in general, fish contains much more TMAO than you're going to produce from just eating choline or carnitine. Then, if we start using it as a surrogate outcome, we have to start to explain why fish is not associated with cardiovascular disease risk and it's actually associated with less, which you can try to make that argument. It just does, it's like a huge amount of mental gymnastics to come up with why fish is sometimes protected, but then confounded, and why we should still restrict other sources of choline.
So this is one of the most interesting aspects of this whole TMAO story. People who claim that TMAO causes disease and that we want to avoid foods that raise TMAO for heart health. But as Kevin pointed out, fish and whole grains, for example, can raise TMAO levels, and yet they're tied to better cardiovascular outcomes. People who eat more fish and more whole grains have less cardiovascular disease, better cardiovascular health in general. So it's a very difficult argument to make.
And you do start to get into this issue that I think is most compelling, is that choline intakes are relatively narrow within the population. You have people consuming 200 to 400 milligrams a day for the most part, with some tail ends there, but TMAO levels can vary quite a bit. And so there's not a lot of room to budge in dietary intakes of choline to start to really decrease intakes and then hope to lower cardiovascular risk. And you have to ask yourself, at what point you decrease intake so much that you start to increase the risk of signs and symptoms of deficiency, which that might have a counterbalancing effect for disease risk, but also just quality of life. And the data is just not there to start to put a marker on, if people aim for this intake, they'll reduce cardiovascular risk, they'll still have a low risk of deficiency. And so I don't foresee any dietary guidelines anytime soon saying restrict choline for reduced cardiovascular risk, but I might be wrong, but who knows?
It's an interesting point. The the fish and the whole grains and all these foods that are associated with better cardiovascular outcomes, if they lead to a strong elevation of TMAO, it would be an argument against TMAO being a positive cardiovascular concern. I guess the devil's advocate there would be that it's a net effect thing. So it
Yeah.
is causing some kind of harm, but then other components of the same food outweigh the harm. The omega-3s in the fish or something like that are so good that it outweighs the harm of the TMAO
Yep.
possibility. But
Yeah, this is why there's no framework that anyone has universally accepted for how to assess an individual food's risk on cardiovascular disease. Even like the saturated fat content is probably the best we have, but still controversial. And there's food source variability, and what you replace it with matters a good bit. And so, uh, we have basic guidelines, but they're almost all predominantly driven by really controlled metabolic studies and their effects on a few surrogate outcomes. But I think the weight increasingly has been placed on observational studies with disease endpoints. And there, the mechanism doesn't really matter. You're just looking at whether the food associates with the disease endpoint. And there are, issues in interpreting those and how much weight you might want to put on them. And there's lots of critiques around our potential over-reliance on observational data. But at the end of the day, whether we add TMAO to the list of surrogate indicators alongside blood pressure, blood LDL cholesterol levels, maybe blood glucose, bone mineral density, the very few surrogate indicators of chronic disease risk that we have, whether we want to add TMAO to our risk calculus, I don't, no panel has decided that we should do that yet. The FDA doesn't look at it as a surrogate to guide health claims. And so, I think my final caution before I have to run here is that that there's always early science. You can look at every single micronutrient. I'm editing a textbook right now on all the nutrients, and every single area has a little bit of evidence of like, you can get really excited and say, oh, maybe this nutrient is related to this disease outcome in a good way. But every nutrient also has some early evidence that maybe this nutrient is related to this disease in a bad way. And almost virtually for all of them, we don't have cardiovascular endpoint trials or chronic disease endpoint trials, or even basic 6-week trials where we look at quality of life indicators or anything. And so you can get super hyped about individual nutrients and individual biomarkers, but that can often miss the forest for the trees. And if you try to apply that standard equally across all nutrients, you'll come up with the most hodgepodge mix of, well, this nutrient might be bad, this nutrient might be good. You have no confidence in the dose at which it might be good and the dose at which it might be bad. And so I take a big, part of why I'm in nutrition research is because there's so much research that needs to be done. But keeping the big picture in view and not getting hyper focused on individual nutrients with individual single biomarkers that you think are like the one biomarker to rule them all.
So this is a key point. When we look at isolated nutrients, we can always come up with some story for why that nutrient is really good, really health promoting, or really bad, really toxic for us, based on experiments like test tube data or animal model experiments. Basically, these things that we call mechanisms. And we see this on social media all the time. One guru will tell you to avoid eggs because of TMAO, or dairy because of IGF-1, or legumes because of lectins, or soy because of phytoestrogens. But we could take each of those isolated nutrients and flip the script very easily with just different test tube or mouse data. That's almost always doable. So at the end of the day, what really matters is the net effect in human beings eating the foods, much more than these stories based on lab experiments. And when you understand that, 95% of the stories on social media, you can see right through them.
And also, I think staying humble, like the evidence, the effects are really small, even for things that we think are important. Like dietary sodium's effect on blood pressure is relatively small. It's the net effect of adding in all the things in a DASH diet beyond just sodium that additively become like a first-line pharmacological blood pressure measurement. But I think we should have strong public health campaigns around like reducing sodium intake and not. But you would also, for yourself and working with an individual, There's no like one thing in the DASH dietary pattern that I think like absolutely trumps it. If you can make progress on a few things, that's great as a clinician. And if you don't ever reach the goal where you hit all 9 things that the DASH diet aimed for, that's fine. Like that patient is not going to drop dead tomorrow. So incremental changes, acknowledging that most nutrition effects that we can even measure are incredibly small. And if we're not seeing them or we're uncertain about what the changes mean, like there's plenty of other things that you can make meaningful, you can put your effort into.
I think that's a good point and kind of a good take-home message to wrap up, because even if the effect of TMAO is TBD and we're not sure if it's direct cause or just a surrogate, I mean, if the evidence for a specific food, whether it's fish or whole grains or whatever it may be, is consistently positive, whether it acts through TMAO or some other mechanism or a mix of mechanisms ends up being secondary. At the end of the day, what we want is the outcome. Negative
Yeah.
or positive, we want to know what the net health effect of the food is.
Exactly.